Welcome back. This is Chapter 7, Lecture 2. We have made it on to surface area and volume ratio, or surface area to volume ratio, and we have in the last lecture talked about the cell theory and all the people that contributed to the cell theory. Remember, the three components of the cell theory is the following. All cells come from pre-existing cells. Cells are the basic unit of life. And all living things are composed of cells. Now, I said those in reverse order of what we typically learn them in, but order really doesn't matter. Okay, it's all living things are made of cells. Cells are the basic unit of life, and all cells come from pre-existing cells. Then we talked a little bit about five fellas that were instrumental in the development of that cell theory. We started with Hooke and how he coined the term cell, and so all of cytology and all of the cell biology that we know of today is really kind of started with him. Went on to Leeuwenhoek, and he was the one that was actually able to use a compound microscope to observe real life living cells and real life living microorganisms. He discussed those microorganisms as animalcules. Schleiden and Schwann, remember? Schleiden is the dude that said all plants are made of cells because he was a botanist, and he looked at a lot of plant tissues and noticed that it doesn't matter what type of plant you are, all plants are made of cells. And then Schwann was the dude that said all animals are made of cells. And remember, you can remember that by saying Schwann is a majestic goose. And I don't know if you've ever seen a swan, but they're a pretty magnificent beast. Okay, they look very, very calm and tranquil until you get close to them, and I've discussed in my classes, at least with some students, that I do have a story in which I was chased off of a nest from a nesting female swan and or nesting female goose, and let me tell you, they're fast and they bite. And so if you see a swan or goose that looks like they're sitting on a nest, leave them alone, because they're faster than they look. Okay, they lower that neck and they charge like a battering ram. But anyway, I'm going to start Chapter 7, Lecture 2 with a little kind of scenario, and I've got this little drawing here. And in this drawing, we have a bunch of little tiny happy faces, and we also have a big, scary, sad face. Okay, and so these little faces that are happy say ha 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 ha, and the big scary sad face says I'm starving. And so my question to you is, why would I have this picture on here? And I want you to pause, think about it, and then we'll talk about it. Did you pause? Okay. Good. So you should have had time to think about it. And so when we talk about little cells versus a big cell, or little faces composed, or a bunch of little faces instead of a big sad face, is this whole idea of cell size and the idea of surface area to volume ratio. These make up way smaller volume, or make up a way smaller volume than this one. This one is huge volume. Okay, and you can easily understand when you kind of, and I'm trying to color or highlight the inside of this with a mouse, and so I'm not very good, but if we highlighted all of the interior section, and now it looks like a big yellow scary face, but if we highlight all of this in, this would be the volume that this face or cell makes up. And if we compare that much yellow to this much yellow, so much smaller, so much less volume, and so I guess at this point we haven't talked about surface area or volume or the surface area to volume ratio, but I want you to kind of just think about how much volume there is here versus how little volume there is there, and then we'll get into kind of the specific parameters of surface area to volume ratio, some of the limits to cell size, and some of that things. So hang on, stay tuned. So when we talk about cell size and cell function, remember that cell size cells are not of one size. There are very large cells and there are very small cells. A lot of bacteria, for instance, are very, very small. Okay, and so look at this little laser pointer I have here. Right, cells are not of one size. Bacteria are very small. Some eukaryotes can be very big. They range from microscopic in size to the size of an ostrich egg, and I know the entire ostrich egg isn't one cell. There's actually kind of one disc cell that's inside that egg. And so when you think of ostrich egg, don't think of the entire thing as being one cell. But there are muscle, or there are nerve cells that are up to like a meter long, which is like 3 feet. Okay, there are some giant algae that are several meters long, and so these very large cells have to have a way to support themselves. And when you think of a cell that is large, they have to have more specialized support systems. When a cell is really small, it doesn't have to have as much support because things diffuse in and out at a much quicker rate, and so it's a lot easier for them to survive. Longer cells must have a way to transport nutrients. Remember, if a cell is large, it has to transport the nutrients and minerals all the way through the cell. And so if you're a small cell, you have to transmit and transport those things a very short distance. I don't know about you, but I think it's a lot easier to transport things or carry things a short distance than it is to transport or carry them a long distance. Okay? Just my personal opinion. So here is a little bit of further information, and we talk about these pictures. Okay, this one is a very large cell, and this one over here is a same size image, but instead of having a cell that's 30 micrometers by 30 micrometers, let's say it's a cube, and I know it doesn't really look like a cube, but if we have 27 smaller cubes that make up this one large cube, the volume is the same. The only difference here is the total surface area is much bigger, right? If we looked at the surface area of one big cell, it's 5,400 micrometers in this case, and that would be micrometers squared, because remember, we're looking at a specific surface area. It's a 3D shape. And then the total surface area of these 27 smaller cubes would be 16,200 micrometers squared. That's a huge difference, right? And we didn't change the volume. And so right here is kind of what we're summing up. It is the cell surface to volume ratio that determines and limits the cell size. Okay, and I say that all the time. It is the surface area to volume ratio that determines the limits of that cell size. It isn't the amount of organelles it has. It isn't the amount of DNA it has. It isn't the amount of proteins it makes. It isn't the amount of distance it can cover by whipping its flagella or anything like that. It is literally the amount of surface area and the amount of volume. When you make a ratio, it has to be efficient enough and it has to be small enough in order for this cell to actually maximize all the nutrients and minerals it has. Right? So, does this make sense? If we have a 30 by 30 cube, it has this much surface area. And if you take that exact same cube, but we just divide it up into 27 smaller components, because now we have all these sides that are on the interior of the bigger cube, we have all these sides that's adding to or just multiplying the surface area. And so we get a much larger surface area. And when we talk about cell membrane, surface area is key. Okay, here's another way to look at it. Again, if we have 1 centimeter by 1 centimeter by 1 centimeter cube, you have a surface area of 6 centimeters cubed, a volume of 1 centimeter cubed, and that gives us a surface area to volume ratio of 6 to 1. Okay, that's really good. That's really high surface area to volume ratio. We want more surface area and less volume. If you increase that to a 2 centimeter by 2 centimeter cube, then the side length doubles, and so we have 24 centimeters cubed now, but the volume we have 8 centimeters cubed now, and so that automatically lowers our surface area to volume ratio to 3 to 1. Okay, so just by doubling the length of our side, we have decreased our surface area to volume ratio by half. Okay, or we've cut the ratio in half. And then if we add another 50% to our cube, meaning we add 1 extra centimeter to all sides, now we have 54 centimeters cubed, our volume is 27 centimeters cubed, and our surface area to volume ratio now is 2 to 1. Okay, this one is much more efficient than this one, which is much more efficient than this one, and 6 to 1 is way more efficient than 2 to 1. Why? Because we want surface area. Surface area is where all of the endocytosis and exocytosis takes place. It's where the oxygen and carbon dioxide diffuses in and out. It's where the cell gets food. It's where the cell gets oxygen. It's where the cell gets rid of wastes. It's really, really important. Here's another way to look at it. If we look at a radius, and I have 3 different sizes, maybe we have the first cell, the second cell, and the third cell. The first cell has a radius of 1 times, okay, or 1x, so this could be a variable. Let's just say it's 1, this one is 2, and this one is 3. If we compare these 3 different size cells to the surface area, our surface area increases from 1 to 4 to 9. That's not a very big jump in our surface area. Our volume, however, increases from 1 to 8 to 27. And so which one increases faster? The surface area increases, but the volume increases faster. Okay, and so this is volume, this is surface area. Surface area increases as we get bigger, but the volume increases faster at a faster rate. And so if the volume, which is our denominator, is going to increase faster than our numerator, it's going to decrease that ratio. So what is it all about? Cell size and function. A cell's efficiency is determined by its size. Simple. Right? You cannot be too large if you're a cell. Why are the biggest animals, i.e. blue whale or elephant, why are they composed of more cells and not just bigger cells? Because if you looked under a microscope at an elephant tissue, the cells that make up an elephant or cells that make up a blue whale are not any bigger than the cells that make up you, and they're not any bigger than the cells that make up your dog. Okay, and so why would an animal that is 3, 4, 5, 10 times bigger than you be made of the same exact size cells? And the reason is that the cell's ability to survive is directly dependent on its size. Okay, the cell cannot be too big or too small. It has to be just right. It's like that Goldilocks. Okay, there's a bed and there's like 2. Well, anyway, I don't know. But you've got to be able to have just right, not too big, not too small. And so what are the advantages of a cell size or a cell surface area to volume ratio? That's where all of our nutrients are exchanged. Okay, if this is the cell surface area, all of our nutrients are going in and out. Our waste products are going out, and our oxygen's going in. Okay, all of these things happen at the surface. And so if you have a very large cell, these nutrients have to travel this far to get all the way to the center. Now they're coming in at all different directions, but the arrows are the same size. If we have a cell that's this size, the arrows are half or a third or a fourth the length. Okay, it's much easier to get nutrients into a small cell than it is to get nutrients into a big cell. Faster doubling times, again, it's easier to split this cell in half and produce 2 cells through cell division than it is to cut this cell all the way in half and produce 2 cells. Okay, so faster doubling times, it's easier to exchange nutrients, and we can have larger populations with greater mutation, which lead to greater survival in the same amount of space. Okay, so think about bacteria populations. If you were infected with some bacterial population that is pathogenic and it causes you to be sick, is it more likely that bacteria is going to survive in you if it's a larger population or a smaller population? I would hope you'd say larger population, right? Our white blood cells can really do a number on populations of bacteria, but if the populations are larger in the same amount of space, there's just by chance greater variation among all those bacteria cells, and just based on natural selection and the mechanisms that drive evolution, you're going to have greater survival of that bacteria, leading to you becoming sick and the bacteria cells kind of winning that battle and surviving. Okay, so larger populations give the population a greater chance of mutation, which leads to greater survival. I mean, this can be easily understood in terms of humans as well. If a human population is larger, then we have a greater chance of mutating randomly, and we have a greater chance of survival. This just shows you the same thing that I drew. And so if I can get this eraser and kind of erase some of this stuff, we can look at these diagrams. Right. This shows you kind of what I was showing you before, only it's a little neater and easier to read. If this is the arrow, and this is the arrow to get from the very center of this cell out, you can tell that this arrow is shorter than this arrow. So nutrient exchange is going to be much faster doubling times in this size cell, and you can fit more of these types of cells in the same space it takes for this one, which leads to greater mutation, greater survival. Okay, so what is this surface area to volume ratio and diffusion? Okay, so this is a lab that we may or may not do, but this is a lab you might see in college. And this is a nutrient agar cube, which is basically like a nutrient jello, which would taste really bad, I'm sure, but bacteria love it, and so we can use it to form cubes at different sizes. And so let's say this is a 1 centimeter by 1 centimeter by 1 centimeter cube, and this is a 2 centimeter by 2 centimeter by 2 centimeter cube, and this is a 3 centimeter by 3 centimeter by 3 centimeter cube. You can see, based on what is pink, what hasn't diffused, meaning and the way this lab is set up, we take these cubes, we soak them in a sodium hydroxide solution after adding phenolphthalein to our agar, and phenolphthalein is an indicator, and it turns pink in the presence of a base. And so when you soak these cubes in a base, the entire cube will turn pink. We can then take these cubes, drop them into something acidic like really weak or really strong HCl, hydrochloric acid, or we could do a sulfuric acid, or we could even do acetic acid like vinegar. And the longer they're in the cubes, the longer the acid diffuses into the cube. Now, we just learned about surface area to volume ratio, and so what can you tell me by looking at these cubes? This one has a greater surface area to volume ratio than this one, and this one has a greater surface area to volume ratio than this one. And so you can tell the smaller the cube, the less of it is pink. Why? Because it has less volume in relation to the surface area. Okay? These are very short, thin margins. These are very thick margins. In fact, in the same amount of time, let's just say this was a 10-minute soak. Okay, we put all 3 of these cubes in at the exact same time, and we let them soak in an acid solution for 10 minutes. Almost within 10 minutes, we had even diffusion all the way through. We didn't have anywhere close to even diffusion throughout here, but even in this smaller cube than this one, we had more diffusion through because it has more surface area and less volume in relation to this one. So that's all we got for surface area and volume ratio. I'm going to leave you with a little joke. Looks aren't everything. It's what inside you that really matters. A biology teacher told me that once, right? And I'm sure you've been told that before. When you think about surface area and volume ratio in cells, it's really that simple, folks. Right? It's inside that really matters, and I'm the biology teacher that told you that. Peace.